Transposable elements, also known as jumping genes, are DNA sequences that can jump from one location in the genome to another, and they make up about half the genome, but they can create or reverse mutations, which can affect the size of the whole genome. There are two main types of transposable elements, including DNA transposons, which move through a cut and paste method, and retrotransposons, which move through copy and paste and also use an RNA intermediate. DNA transposons have terminal inverted repeats, which transposase, the enzyme that it cuts the transposon, is able to bind to and cleave the transposon. The transposon is then integrated into the target site in the genome, and DNA repair fills in the gaps. What was left over from the cleavage of the transposon is then repaired and left as a transposon footprint. Transposons do not use RNA intermediates, which is important to note. On the other hand, retrotransposons start in the nucleus and they have long terminal repeats and carry genes GAG and PAL, which are essential to complete transposition. They get transcribed into an RNA intermediate, which then leaves the nucleus to go into the cytoplasm, where the reverse transcriptase enzyme makes a DNA copy of the RNA using reverse transcription. But reverse transcriptase does not have any proofreading abilities, which increases the likelihood of mutations. The new DNA intermediate is then sent back into the nucleus, where the copy can be integrated into the target DNA in a new chromosomal location. TEs stop jumping around the genome once it has managed to repress transcription of them through these processes. Genes have some key regions, including a promoter, exons, the coding regions, and introns, the non-coding regions. Transposable elements can have different effects on genes depending on where in the genome they jump into. If they jump into an exon, a frameshift mutation will likely occur if the insertion isn't a multiple of 3 nucleotides. If they jump into a promoter, this can enhance or repress the amount of transcription. However, if they jump into an intron, this will most likely have no effect on the protein it codes for and the amount of expression. Though transposable elements can cause mutations, they can also create genetic diversity and they drive evolution, and they're constantly shaping our genomes. Thank you for watching. I hope you enjoyed.